today, and it's very wonderful to be here with you guys and to be able to discuss and share some knowledge on Phage work and Phage research as well. And today we have a very, very interesting topic and a very interesting topic, as well as we have a very one of the pioneers in Phage research right now. She will be taking us through some of our work as we continue to learn. But in the spirit of welcoming each and every person, I would like to ask you a very small icebreaker as well, just to keep everything in light moments as we continue. Earlier today, I think just a few minutes ago, one of my friends mentioned that or asked me a question, asked some of us a questions like, why do we celebrate Phage Week, especially on 22nd October, right? And I'd love to hear from you guys. Why do you think we celebrate Phage Week from 22nd October? It's a very curious question and a very interesting one. Maybe you can just mention it on the chat or unmute and tell us something. All right. Maybe I'll give you a hint. We all remember that Frederick Schwartz is one of the pioneers who discovered Phage work, phages, bacterial phages as well. And on 22nd October is usually his birthday, right? And I think it is very interesting that we get to celebrate Phage Week starting from his birthday, and we are able now to build on it and yes, share some knowledge around this, around the facts, around bacteriophages, and even so many things that come in between, right? So that is one interesting thought that you can be thinking of as we continue to have a discussion on today's topic. Yes, Dr. Tobi.
I can add an interesting story about Frederick Schwartz in that Dr. Martha Clokey, who many of you know her work from the UK, she's the senior instructor for our Phages for Global Health laboratory training workshops. She was invited to speak at a school in the UK. I don't remember exactly where it was, where it turns out the headmaster, I believe it was the grandson of Frederick Schwartz. And so she enjoyed having that connection with him.
Interesting. That's very interesting. Wow, that's good. Yeah. Yes. So we all see that there are so many interesting facts around this, and it's lovely to continue to dig a little bit more and get more facts and have some little light moments as we continue. And thank you again, Dr. Tobi, for that. So I'll jump on to today's session, and today we have a very interesting topic and one that we cannot go by without, right? So, but before I go any further on that, I'd love to introduce my fellow moderators around the panel, then we can move on from there. So number one, I'd like to start by introducing Nelson, who is a lead advocacy lead for the Phage hunters, and he has been able to be the pioneer of enabling us to have this session and preparing all this session that we are going to have today. And thank you, Nelson, for that. We also have Nilofa Shahik, who is part of the Phage group, which is a website. You can be able to find a lot of science communication dedicated to exploring the fascinating world of bacteriophages, viruses, and you see on their pages, you'll be able to see even opportunities around bacteriophages research and everything. So you will also be able to look on their page. It's called the Phage.xyz. So please check their page as well. We also have Raphael with us, also part of the Phage group. And as I've mentioned before, the main aim is to be able to communicate science around the bacteriophages. So please be able to check their page, and it's a pleasure to also work with them. Thank you, Ellie, for sharing their page. So please visit their page as well. And my name is Elkana Moody, and a member for the advocacy group for Phage Hunters. And it's also a pleasure to have you with us here and to be with you through this session. I'll be guiding you through this session. Thank you again. So let's just jump in right into today's topic. And today's topic is one of interesting one because we all know that the science of phages is advancing very fast, perhaps even faster than any of us expected. But yet, we found that as innovation accelerates, the regulation often lags behind, right? So the challenge we face is simple but serious. And we find that we ask ourselves, how do we make sure life-saving phage therapies reach the patient safely, quickly, and equitably without being trapped in the outdated system, right? So our session today is designed to unpack exactly that, not in theory, but in reality. And we look at what's working, what's missing, and future might look like, especially in the low and middle income countries. Nice. So I will take this opportunity to welcome a very important speaker for us today. And it is my great honor to introduce someone who's worked at Tobi Nagel, and an organization that's been pioneering the use of phage technology to combat antimicrobial resistance in the global south. And through her leadership, Phages for Global Health has trained hundreds of scientists across Africa and Asia, being able to bridge research and application in a truly transformative way. And it is a testimony that I myself have also been part of that, and it is really good to have been a part of that group as well. And what I personally find most inspiring about Dr. Nagel's work is that her focus is on access and equity. And she's not just asking about how phages can be developed, but how they can be able to reach the people who need it the most. In the regions where we find that antibiotic resistance is a daily reality. And today, she will take us through her journey, her insights, her reflection, and reflections on the current regulatory landscape for phage therapy and how we might be able to design more adaptive and inclusive framework going forward. Please join me in welcoming Dr. Tobi. And yes. Dr. Tobi, the floor is yours. Thank you.
Thank you for having me, and it's nice to see all of you here and see your names here. Thank you for that kind introduction. I am going to share my screen. now. So can you see my screen in full screen mode? Oops.
Yes.
Can you still see it?
Yes,
No, no.
yes.
Now we can, yeah.
And you can see my arrow?
Yes.
Perfect. Well, thank you. It's a pleasure to be with all of you today. Thank you from, I know that you're from all over, not just Africa. So welcome to everyone, and thank you for having me. As was mentioned, I lead Phages for Global Health, and we do what we do because of this data shown in this graph, that AMR, antimicrobial resistance, is expected to kill 10 million people each year by 2050. And unfortunately, that means that 90% of those deaths will occur in Africa and Asia. And most of the phage experts are elsewhere. I would say they're also in other parts of the world too, not just Europe and the US, but the majority are in Europe and the US. And so, simply put, the mission of Phages for Global Health is to bridge that gap by empowering phage product development in Africa and Asia, specifically for those regions. And so, as part of that work, I am currently doing this Fulbright Global Scholar project where I'm gathering information about how phages are regulated in high-income countries, focusing primarily on where that's being done in the US, Europe, and Australia, and sharing that with national leaders in low-income countries. But there's three types of information that I've been gathering. One is formal guidelines, of which, frankly, there aren't that many, and I'll talk about that quite a bit. But informal guidance, for example, the US FDA has presented a fair bit at conferences. I was actually at a conference in Washington, DC last week where they presented, and where I talked a fair bit with 3 people from the FDA who are very, very open to communicating, similar from other places in Europe. And also, I've done a fair bit of interviewing people who typically are with companies, but sometimes with academic phage centers. And asking what's their experience. Oops, I think we need to mute somebody. Asking what's their experience of bringing those phage products through the regulatory systems. What's their actual practical experience on the ground? So as I said, I'm sharing that with regulators in LMIC countries. Specifically, Fulbright is funding 2 months each in Malaysia, Kenya, and Uganda. I've already finished the 2 months in Malaysia and Kenya. I believe I met Ellie and the team during my 2 months in Kenya, which was 2024 spring. And then I was in Uganda last month for 1 month, and I will go back to Uganda for the final month beginning in mid-March. And the overall goal here is to benefit from lessons learned overseas, but really support your regional regulators in developing guidelines that are relevant for your regions. And it might be different, and frankly, I think the desperation and the need is much greater in your regions already. I think you already have higher rates of antibiotic resistance probably than we do in the US, for example. And so that frames what I'm going to talk about today. So let me start by highlighting that clinical trials do not require formal phage guidelines. And that's a myth that's often out there, that you need your country to have formal phage guidelines before you can move forward. And the reality is that the last time I checked, at least 35 clinical trials had been initiated in 16 different countries. And I can tell you that most, if not all, of those countries did not have formal phage guidelines when they started those clinical trials. They just started by communicating with their regulators and figuring out what would be their path forward. For example, the US has no formal guidelines yet, still, And yet I believe more clinical trials have begun here, and possibly more compassionate use cases have been done here. Basically, what most countries do is figure out how to fit phages into their existing regulatory systems, and they typically consider phages to be biological products, which makes sense. And they are just treated as other biological products. Now, we know that there are certain data points that are relevant to phages that are not different, relevant to other types of biological products. And so there's sort of a standard set of data that most regulators now expect to see when reviewing phage products. So I'm going to talk about what those standard data sets are, and they're pretty much the same for all three types of pathways that can be compassionate use pathways. Compassionate use has different names in different countries. I'll talk about that. Could be through clinical trials, or it could be through the magistral, or sometimes it's called the compounded phages approach, which at the moment is officially approved in Belgium and Portugal, and it's also part of the European Pharmacopoeia, which is a set of documents that gives guidelines across all of the European countries that are part of that. And so theoretically, the magistral approach could apply across Europe, but ultimately it's up to each country to decide if they want to do that or not. Belgium and Portugal are the only ones, again, who have formally done this. But I believe in, for example, the Netherlands, they tend to follow the European Pharmacopoeia, which is basically the magistral system. So let's start with the US FDA, Food and Drug Administration, which is the regulator in the US. And they have something called their Single Patient Investigational New Drug, or IND, SPIND. And if you're not familiar with clinical trials, investigational new drug, or IND, is usually the application you would apply to start those clinical trials. That's the name of the application, IND. For compassionate use, which is single patient IND, in the US, that's officially called expanded access, or they often refer to it as compassionate use as well. And so they have while the US FDA does not have any formal guidelines, they have put out this relatively short document called the Single Patient IND Request Shell document that gives you an idea of the information they want to see. And this is just an optional tool, and you still have to fill out. There are specific forms that need to be filled out for compassionate use cases for the FDA. And I think most countries, I seem to recall when I was in Kenya, Kenya does have a compassionate use process. So there are specific, I think, online forms you would need to fill out for that. But in general, what they're looking for in this document is three types of information, or things that fall into three categories. One is personal information, both about the investigator and their qualifications and the patient. And also they want to see what's the form you're using to get informed consent from the patient, and also the IRB, or Investigational Review Board, approval from the institution where you'd be doing the treatment. So that's all makes sense. You need that personal information. Then you want the clinical information. What's the disease in this patient that you're treating? What's the rationale for using phage therapy? And what's the proposed treatment plan, with the understanding that that treatment plan might need to be modified as you go through the process. And then finally, and what I'll focus on more today, is the phage information. What exactly do they want to know about that phage and how you've characterized it? And really it comes down to 6 data points that they pretty much all regulators want to see, whether it's for compassionate use, clinical trial, or even the magistral phage approach. And let me start by saying you have a preset cocktail or a phage bank. They typically have flexibility in what they will allow, with the understanding that all of those phages have been characterized with the following 6 data points. 1, you've shown that they're strictly lytic. That's typically done by genomic evaluation. And they want to see data not only for the phage, but also for the bacteria that you use to produce the phage. And in particular, they want to see lack of toxins, AMR genes, virulence genes in both the phage and the bacteria. And they want to see also lack of prophages in the bacteria that is used to produce the phage. Then, of course, you need to show that your phage would have some kind of activity, even on this, either on the specific strain, the patient's strain, or on the general type of pathogen that you want to target, say salmonella. They definitely want to make sure that it's free from any impurities that might come from the bacteria that were used to make the phage, possibly from chemicals that were used to purify the phages. I'll talk about that a little bit more in a later slide. And this purity typically is done, you have it done by an outside organization. So outside your own lab, there are labs that officially say, yes, this is a sterile product, and you need to show documentation of that. Or they might look at endotoxin and show documentation of that. You also need to show that for as long as you're going to be needing to store that phage at whatever temperature you're storing it at, it is stable. And if it's a combination of phages, you need to show that they're combined. When combined, they're still stable for as long as you're going to be storing them. All of this makes sense, right? And then the sixth data point, there is slight variation amongst regulators whether you really need to include transduction data or not. And this would be looking at generalized transduction, not specific transduction, in terms of the capability of that phage to transfer a gene, say, to a bacteria. And you definitely don't want that to happen, so you want those rates to be quite low. And often what they use is, say, GFP, bacterial gene biomarker, and see if the phage transfers any genes. There is a lot of debate about whether or not to include this, and frankly, how to measure it, because it's difficult to measure it in a way that might truly represent what's happening in the body. So that's kind of the biggest debate point for that. Having said all that, the US FDA does require GMP manufacturing for clinical trials, not for compassionate use cases. And even for clinical trials, they have what they call a sliding scale for phase 1. And, you know, clinical trials have phase 1, 2, and 3. And for the early stages, they say, well, you're still figuring out your manufacturing conditions. So as long as your product meets the 6 data points above, maybe it doesn't need to be done by GMP manufacturing. And the website I'm showing in the lower right here, fda.gov website, has a document that specifically talks about their GMP sliding scale requirements. And then the publication from 2017 also gives some interesting and helpful overall guidelines, even though that's a few years old now. Excuse me. So that's what the US FDA wants to see for the phage product. Then they also want to see specific details for your clinical trial protocol and processes. For one thing, they would love it if you pre-specify as much as possible how you're going to deliver that phage, what the pathogen is, what the site of infection site is. But actually, you don't have to pre-specify all of that if there's going to be a need for flexibility. Say you're going in with, you want to target Pseudomonas, but that might be at multiple different infection sites within the body or something like that. Or maybe you start with inhaled phages, but you decide you need to add intravenously administered phages. That's typically allowed, as long as your phages are really purified. Then they want to make sure that you've really made sure that you've got appropriate inclusion and exclusion criteria for your patients, that your investigators, you've documented that they're qualified and that they can appropriately manage anything that comes up with this study. that you have predefined adverse events. What would that look like if a patient undergoes an adverse event, and how are you monitoring for those? And in fact, based on those, you might stop treating a patient or stop the entire study if you have too many adverse events. Now, we know that that's typically not the case for phages if they're appropriately characterized and purified, but nonetheless, all clinical trials worldwide pretty much need all of these requirements. And then, of course, you need IRB approval, institutional review board approval from your local institution, informed consent from the patient, and you need to keep the patient's information confidential. Now, this is something interesting that was published by the US FDA. They pointed out that often in phase 1 clinical trials, you're doing what's called a safety study. But a safety data may not be relevant in a healthy patient who doesn't have the bacteria in their body, because then you won't get amplification of the phage in that bacteria. And so maybe they suggest you could consider doing your safety study, your phase 1 study, in infected patients rather than in healthy patients. Though we all know that one of the values of phages is that they don't alter the microbiome, and there might be value in documenting that. So you've got colonized patients. Many of us are colonized with Staph aureus in our noses. But if you could show that you could treat for that, for example, without altering the microbiome, that could be beneficial. So that was for the US side. Now let's talk about Europe. And you're probably familiar with the European Medicines Agency, or EMA, which was established to, quote, harmonize but not replace the work of national regulatory bodies. And ultimately, the national regulators have immediate jurisdiction about what happens in their country. So France gets to decide, even if they might go against the primary recommendations of the European Medicines Agency or other countries, for example. Now, I want to highlight that in Africa, the Africans Medicines Agency was initiated in 2019. I believe it was formally ratified a couple years after that, and they're still being set up now. And the idea is that they will follow a similar model as the EMA. I think it's a great idea. And the first director general was just appointed a few months ago. She was the former head of the regulatory agency in Ghana. So it's exciting to see this process moving forward, though I don't think it's fully implemented or in place yet. So the process that the EMA comes up with when they want to develop guidelines is they start with a concept paper that they put out for public consultation. It's often about 4 months, where they ask for people to give written input about what the guidelines might be, in this case for phages. Then they review that public input and then publish the guidelines. And so they first did that for veterinary applications of phages. They had a 4-month window in 2023 for people to give input about how phages for veterinary applications should be regulated. And then they officially published those guidelines later that year. And then for human applications of phages, the concept paper was out in that window closed in early 2024, and it's now under review. And so that's taken longer than the veterinary. And we all assume that's because there's more debate going on. But we're hoping that the human guidelines are not too different from the veterinary guidelines, because they're fantastic guidelines to be in my opinion. And let me tell you what those guidelines are. Again, the required data that they have for each phage are the same 6 data points as you saw for the FDA. Lytic, look at the genome of both the phage as well as the bacteria, potency or host range, purity, stability, and they do want to see transduction data. They also mention that you could add, but it's not required, information on the activity of the phage on biofilms, whether there's antagonism or synergy with antibiotics. And if you know anything about the resistance frequency or mechanisms or time frame, sharing that and maybe building into your cocktail some phages that might be able to overcome some of those mechanisms, for example. And then again, for the bacteria itself, they want to phages, toxins that could be detrimental. What I like, though, is even the text specifically says, if freedom from these elements is not possible, justify it, basically. Justify that it has no negative effects. So again, they're willing, basically have a discussion with the regulator is how you can read that, which is what we should always be doing as drug developers, is having an ongoing dialogue with regulators. It's not that they have a checklist. I mean, they do have these data points they want to see, but they're understanding that phages are a novel type of drug and that we need to have discussions about these. So there's some overarching themes I want to highlight from these EMA veterinary guidelines for phage products. And the first is that the phage-host interactions are dynamic, so the regulations should be too, should be adaptable. Isn't that interesting? And I'm going to give you a number of quotes directly from the guidelines. A flexible composition of phage products is expected to be the usual situation. Manufacturers may pick monophage or single phage components, single phages, from those included in the approved dossier to match the geographical distribution and phage resistance patterns of targeted bacteria in different countries or for different individuals. Different compositions of the parental product can be marketed at the same time or at different times in the same or different countries to address different epidemiological needs. That's really quite flexible, isn't it? Essentially what it's saying is you can have a relatively small, let's say, collection of phages that you have thoroughly characterized according to those 6 data points. And then you might want to swap in and out the phages over time, or maybe Kenya needs these 3 phages, but Uganda needs these 4 phages that overlap a little bit, but have some differences because your bacterial strains are different in those countries, something like that. So this is really recognizing the value of that. Now, because there's that flexibility, they really put an emphasis on robust manufacturing process to make sure you've got still a high quality product while enabling this flexibility. So the upstream part of the manufacturing process, meaning the amplification or the actual production of the phages, may need to be changed. For example, you might need a new bacterial host if you have to switch to a different phage. But you want to ensure that the downstream part of the manufacturing process, the purification and the formulation, is robust towards upstream changes in phages so that you still have that quality. So we really need to start thinking about the differences between that upstream and that downstream purification process. And finally, they specifically say that therapeutic phages could be natural, adapted in vitro, or even genetically modified. Not a problem. Genetically modified phages are now going through clinical trials, at least in the US, and I think also in Europe. So those haven't been a concern. Excuse me. And some genetically modified phages have been used, especially for mycobacterial infections in compassionate use cases. So focusing on these purification techniques, so at the Evergreen Phage Conference, which took place here in the US in August, I organized a pre-conference workshop on overview of phage purification techniques because we really need to start focusing on this downstream part and understanding how do you make sure you've really got properly purified phages to put into a patient. And I want to highlight, we had 6 great speakers. The first 2 sort of gave some introductory concepts. And I want to highlight that the last 4 talked about the 4 most common technologies that are used for purifying phages. And these are ones that all the regulators seem to, be familiar with when reviewing phage products. And realistically, the one that is probably going to be most accessible in your countries, at least in Africa, is dialysis. I'm not sure, many of your your institutions don't have ultracentrifuges, tangential flow filtration filters can be quite expensive, and not all institutions have chromatography instruments needed for this. And so let me highlight that we're going to eventually make all of these lecture videos, all 6 lecture videos, available through our Phages for Global Health website. And so if you're signed up, if you go to our website, you can sign up, scroll down to the bottom, you can sign up for our newsletter, and we will announce when those are available through our email newsletter. So I think this needs to become part of the next learning curve as phage products start to be throughout Africa. And let's see, we've got also people from South America and from the Middle East, I believe, on the call. So let me finish by talking about, or almost finish by talking about, the magistral phage system, which began in Belgium in 2018. And let me highlight that this is actually not an entirely new regulatory system. The monograph system was already in place in Belgium. And what that means is there were different monographs that were written for different types of medical drug applications. And if you've got an approved monograph within the monograph system in Belgium, then you can have drugs produced according to that monograph. So what was done here was they wrote, led by Jean-Paul Piernet, Daniel De Vos, and Gilbert Verbeken, and many others. They wrote I think it's only about 2 pages, a monograph that outlines how phages should be produced in order to be given on a personalized level to a single patient. And so under this monograph, what it says is that a pharmacist in Belgium can provide natural phages, not engineered phages, as the drug, as the active pharmaceutical ingredient, as long as 3 conditions are met: that there is a prescription from a physician for that phage, that the patient is pre-named, so this is not just for anyone, but the physician says this patient needs this phage, and then the final product must be approved through an independent government-approved lab, and in Belgium that lab is called Sciensano. And what Sciensano does is essentially look at the same 6 data points, or 5 of the 6 data points that the FDA and the EMA look for, starting with the genome evaluation of the phage, making sure you've got purity, potency, and stability. However, they are very clear in saying this does not need to be GMP produced phages, because you're only making this phage for a single patient, and it just wouldn't make sense to do a large GMP manufacturing batch for 1 patient. And they also do not require transduction data. And as I said before, transduction data is often hard to accurately measure for the condition in which it will be used, so they have chosen not to require that. Now, phages that have been produced through the Belgian magistral system have been exported to at least 12 countries, it might be more now, with in-country approval by the local in-country regulators. And as I mentioned earlier, this magistral or monograph system was incorporated into the European Pharmacopoeia in May of 2024. So theoretically, that means other countries could follow that. And as I said, I'm aware of some doctors and pharmacists in the Netherlands who I believe are working under the European Pharmacopoeia in their country, even though their country, their regulator doesn't have a specific monograph within the Netherlands. And then Portugal officially approved the same magistral system also in 2024. So I've talked about the 3 different pathways for human applications of phages, and I want to talk briefly now about how would this fit into Africa or other countries, lower middle income countries. And let's start by talking about regulatory systems are benchmarked as with having 4 different maturity levels by the World Health Organization. And most high-income countries function at maturity level, or ML4. They have advanced level and continuous improvements. ML3, they're stable, they're well functioning. They might not have continuing improvement. And then ML2 and ML1 have some elements in place, but they're not quite there yet. And so I bring this up because about 70% of low and middle income countries function at maturity level 1 or 2. And so because of this, regulatory systems in most LMICs approve drugs after those drugs have first been approved in high income countries. They basically rely on the regulators in other countries. And so as an example, these are the 9 countries out of 54 in Africa who have reached maturity level 3. None have reached maturity level 4, as far as I know. And in fact, Ethiopia is highlighted here in green because they just reached maturity level 3 weeks ago, I believe. So you may or may not see your country here. The 9 countries are Egypt, Ethiopia, Rwanda, Tanzania, Zimbabwe, South Africa, excuse me, Nigeria, Ghana, and Senegal. And so I think any complication we might have of bringing phages forward is not about phage-specific regulation, but really about these regulators not having systems in place to regulate most drugs, any kind of drug. And so given that, a possible path forward for phage therapy in Africa is to start with compassionate use or magistral phage approaches. That could be valuable first steps because frankly, even in the US and Europe, such cases have enabled the phage producers, the regulators, and the clinicians to figure out how do we work with phages. They are a different beast. So they've used these one-off cases and often have had great results, which has been valuable for the patients, but it's also enabled all these others to learn how to get everything in place. And it might make sense to even treat some of those first patients with phages that have been produced overseas. I know the Belgians are often willing to do that. Jafral, a manufacturer in Slovenia, has told me that they're willing to produce phages for compassionate use cases in Africa and elsewhere. And I suspect that the regulators in Africa and other LMICs would be more comfortable approving the even compassionate use case of application of phages that have already been approved by a regulator overseas, which is understandable. This is going to be their first time working with this new class of drugs as well. But ultimately, we really do need to get these big clinical trial data so that we can have broad applications. I mean, the data in the first slide that I showed says that we're going to have over 4 million people in Africa dying from AMR infections. If we were to try to do that on a personalized approach through the magistral system or through compassionate use cases, that's hard to imagine reaching enough of those 4 million people. But if we can come up with broader applications of phages that are approved through broad clinical trials, that seems to be the most efficient way to reach the people who will need it. Now I'm going to take just a few slides to talk briefly about phages, not for human applications, but for animals, crops, aquaculture. And this is going to vary a lot depending on each country. And I can tell you that it turns out the regulation is not quite as stringent in the US and European countries for phage products that are used in food or in crops. And so what's usually done is they get a sort of a stamp of approval, but it's not the same as the FDA saying you may use this. They can go straight into officially marketing this, even if they haven't shown data that they necessarily work, but they do need to show data that they are safe. And then you can imagine you could add the phages post-harvest, meaning after you slaughter or maybe even on food production surfaces. And the 2 lead countries in the world that have these products are Intralytics and Micreos. Micros is also now working out of Switzerland. And then pre-harvest, the two lead companies that I'm aware of that have marketed products are Omnilytics, which was bought by Phage Lux, and AMP Infotech, which really focuses just on vineyards for wine production. So they're largely focused in my state, in California, where we have a lot of vineyards, with support by a larger Japanese company. Let me quickly talk about Intralytics, the first company here that that I mentioned. Their first product was approved in the U.S. for marketing. It was for Listeria in 2006, so almost 20 years ago. And then they've since had products targeting other bacteria. And they've taken 4 different pathways to get those products approved. And they have this really, I think it's the best paper out there, published in 2013, describing the different pathways within the US that they have used. 3 of those pathways come under the FDA, and there are different categories of how that drug is marketed: as a food additive, as a food contact substance, or what's called generally recognized as safe, or GRAS. I think you've probably heard of that one before. Now, GRAS, I believe, is specific to each phage. So it's not that every phage will automatically get GRAS recognition, certification. It's more that it needs to be, you need to look at the genomic, do genomic evaluation of the phage to show that that particular phage is GRAS, is safe. And then the 4th pathway is under a different agency in the US. It's called the Environmental Protection Agency, or EPA. And there, one of the Intralytics products is marketed and certified as a microbial pesticide. And so I think if any of you are thinking about developing products for non-human applications like these, you really need to start engaging with your regulators to understand which agency oversees food in your country. For example, if I understand right, where I was just in Uganda, I believe it is the National Drug Authority that would oversees phage applications for both people and for food, but not necessarily, it's not necessarily separated out, for example, to include the EPA as in the US. And then let me just show you, I'm aware of these 3 companies that have officially marketed phage products for animals or aquaculture. Proteon, which is based out of Poland, but is selling in multiple countries, has products for poultry and fish, targeting either E. coli, for example, or salmonella for poultry, or targeting multiple bacteria for fish, and they might be given as a feed additive or sprayed on feed or poured into the tank. It varies with each product. Then the next one is in India, and I think we've got someone, I know we have someone on the call from India. Salem Microbes has similar products to Proteon, but targeting, in this case, fish, shrimp instead of fish with Vibrio. Here it's a pond application or a feed supplement, or this product is put into drinking water. And then finally, this Korean company is focused on poultry and swine, targeting some overlapping, but also some different bacteria and feed additives. And there, in fact, if any of you are aware of other companies that have officially marketed phage products, I'd be interested in hearing about those. These are the ones I'm aware of so far. And I'm going to end by just acknowledging a lot of the people who either have hosted me in different countries as we've talked with regulators, or have answered questions from me about how they've gotten phage products through their countries' regulators. And I will end with that. I'll also say that if you go to our website, Phages for Global Health, and again, sign up for our newsletter, you can get more updates from us over time. So I'm going to end there and stop sharing, if I can remember how to do it. Here we go. Okay, and take some questions if we have.
Yes, thank you very much, Dr. Tobi. That has been a very insightful session that you have led us through, starting from understanding the current burden that we have on AMR to all the way through to understanding the overviews of phage purification techniques, and as well as mentioning on the Belgium magistral thing. And you've also taken us through the clinical trial protocol and processes that we need to understand. And it has been a very good session, very insightful one. But maybe as we wait for some of the questions from the participants, I'd like to also bring your mind, or borrow your mind on some of the few aspects that have come across to me. Talking of some of the lessons that we can be able to learn from the Belgian magistral model, or even the recent UK's MHRA guidance. Do you think like this can be following those frameworks? Can they be able to be adapted really well in Africa? Because the way I'm seeing it in Africa is Africa is providing a very big canvas for us to be able to write some of these frameworks better, or even design something that's better yet. So maybe what are your thoughts on that?
Yeah, and I do think that, as I said, I think that's a great starting place potentially to think about personalized phage therapy. I can't imagine doing 4 million personalized phage therapy cases across so many different countries. One thing I forgot to mention, the magistral system is not intended to allow for marketed phage products. So the magistral system will never give a company approval to sell this product in a broad way. It's only intended to enable treatment of individuals. And I think one thing that's going to have to be tightly linked is how are you paying for this phage therapy, as well as how are you regulating it? And I do think that governments are going to need to be involved in this in sort of a public-private partnership, and which is how essentially it's been done in Belgium. It's the military that's largely providing these phages for people. I do think, similar to what we did for COVID, our countries provided COVID vaccines, COVID diagnostics. I think we're going to need our countries to provide, or at least supplement, the cost of phages. And one way to think about it is we need to switch the mindset from how much money can a company make selling phages to how much money can a country not lose to having to manage AMR infections, lost workforce if people are sick, lost children going to school, days of school lost, and lives lost. So it's tightly linked with how are you going to pay for it.
Thank you for clarifying that. And maybe, yeah, as one of the challenges we have globally when it comes to phages is that the definition of phage itself. Like, for example, we find that in the US, it has been approved as a full biologics, right? It has been given a full biological approval, and an example of the same way in Europe, it is in magistral. So maybe and I think this is one of the things that actually it's hard for people and even some of the concerned people around the regulatory and guidance to approve this, because we find that in one place phages can be defined as live, which we know that they are live as an organism, and in other cases uh, they can just be defined as um, forgetting the word, but you get the picture. In other cases, is that how can we be able to find like how can we be able to come up with maybe the right definition when you're explaining this to some of the regulatory bodies? For example, here in Africa, it is so hard to just come and tell someone that I can be able to treat you with a virus or so. You see, it's already building tension in between.
Yep.
Maybe, yes.
Yeah, and so first of all, I think there are many types of drugs that are biologicals. So the US FDA is broken into basically two parts, one that handles the biological drugs and one that handles the chemical drugs. So biologicals, most regulators should be somewhat familiar with that. I agree that virus has a whole different scary sounding meaning, and especially I think virus is a more loaded term in Africa, where you've dealt with not only COVID and HIV, but Ebola and Marburg and other viral diseases. Excuse me. So I actually think it's going to be important to have public engagement activities where we educate the public, not only the regulators, but clinicians, patients, poultry farmers, whatever, about these are safe viruses. These are good viruses that can help you, that are frankly already part of your microbiome. So they're already in you. We're just changing the numbers of them if we treat you with them. So I think there's the public engagement piece. And then I think, personally, I think it's a myth that regulators are not going to be willing to consider phages, because we've already shown that in 16 countries, they are willing to consider phages. And then I think there's another myth that people often get confused by, and that is, as a drug regulator, it's your job to communicate, as a drug developer, it's your job to communicate with the regulator and make the data-based argument for why this drug should be tested and or utilized in patients. And for any type of drug, stem cells, those are a novel type of drug. Those would not have gone forward if drug regulators had not gone to the regulator, drug developers had not gone to the regulator and said, here's why we think you should use stem cells in patients. And here's how we're going about to quality control them. And here's how we those conversations just need to happen. And I think drug developers need to think of approaching regulators with a dialogue, not as a yes or no kind of answer. Because frankly, when people are developing novel drugs, they know the science better than the regulators do. And so it's really up to them to help the regulators understand the nuances. And then the regulators are really smart and can come back with some perspectives that might be really helpful. And so the dialogue should result in a better outcome than either of you would have, either the developer or the regulator would have had on their own. So I would say that as an answer, I guess, to your question. I think you're still muted.
Okay, sorry, I think I was muted. Maybe that's a very good answer that you brought. And I think one of the biggest challenges with phage therapy is that it is it is always evolving. It has its own evolving nature, where phages change, the cocktails adapt. Maybe to hear from your experience, what do you think are the biggest regulatory pain points right now in this case?
And again, I will go back to the slide where I talked about how flexible the regulators are. The EMA guidelines in particular explicitly say you can swap in and out phages as needed for in a product that you call by the same name, even if you swap out the phages. You don't have to do an entirely new clinical trial with each one. And the FDA, when I spoke with them just last week at this conference, they said, yeah, they enable the same thing through their existing structure. They didn't have to make a special structure, regulatory structure to do that. So again, a key message I would give is I think these hurdles, regulatory hurdles, are just myths. I think the conversations haven't happened yet, and the conversations just need to happen. The first people who took phages into clinical trials just went to the FDA and said, hey, can we talk about how we do this? And then they started treating patients after they came to a solution that made sense to both the developer and the regulator.
Okay, great. Thank you. We have a few questions from the audience, and I'd love to read to you. We have a question from Natalie. She's asking, is there any public or private repository of bacterial strains free of prophages, AMR, and virulence genes that are suitable for gene action?
It's a great question. I wish there were. I don't think there are. There are collections of phage strains that are characterized to greater or lesser degrees. And to the extent that they're fully characterized, you might already have that data available. But as far as I know, that has been one of the more challenging things, is coming up with a bacterial strain that is a special strain. No phages or any of those other concerns. And so sometimes I believe some developers have engineered those bacterial strains to have the right.
Okay, thank you. I think, Natalie, I think you have been answered on that. There's also another question from Akihiro. He's asking, now that we have gut microbiome and we all know phages feed on bacteria and replicates more, is it possible maybe that it may have an effect on the gut microbiome, or phages might be able to replicate, creating more concentration in the body, producing an opposite effect on the physiological status of the body or animal?
It's a question, but we know that phages have been in our bodies for as long as humans have had bodies, and we haven't been aware of any negative impacts. Also, I can tell you from the clinical trials that are ongoing, and there's a number of them now, several of them have looked specifically at microbiome and have shown no changes in the microbiome when the phages are targeting pathogenic bacteria, which is what all phage products have been developed for so far. So I don't think that's a concern. I can also say that the FDA and the EMA have both gone on record as saying that they consider phages to be inherently safe because our bodies are constantly exposed to them and have been for millennia. So I don't think we have reason to be concerned about that.
All right, thank you. I think I saw a hand from Raphael. Raphael, you can ask your question.
Oh, actually, it's not a question. Just I work on gut microbiome and virome, and that's why I just wanted to chip in. Yeah, it's a very nice concern from Kiro, but The point is, what we want to remove from our gut is what we are removing, which is a pathogenic bacteria. And that pathogen bacteria actually is causing more harm than the phage we are putting, because it's the one causing destabilization. So once you introduce your phage into the human gut, it will go and remove what is causing destabilization. But if you are concerned by phages that are already inside the gut, most of the phages we have in our gut are actually template phages. They are not lytic. We have a few lytic phages, but we have most of them are template phages, and they help actually most of the bacteria to adapt to the environment in our gut. So we need them for our microbiome to stay stabilized.
Thanks for that
Thank
input.
Thank you for adding that, Raphael. It's really good. It's really good to have people working on the same here. So there's also another question from Mark. He's asking, you recommended that African countries to use imported foreign phages as they develop their regulatory system. How well do phages are isolated in one country kill target bacteria in a different country or continent, considering variation phage bacteria compatibility? For instance, commercial phages isolated in Georgia and Russia showed poor quality, poor efficacy when applied against local bacteria strains in Kenya, but were effective in Europe.
Yep, and I think it really depends on the particular bacteria and phage that you're talking about. And that comes down to always needing to test and make sure that the phages are relevant against the local strains. Very important point. It's one of the reasons that we encourage local phage production and isolation, because I think theoretically, phages isolated locally have evolved to kill the local bacteria. However, there are a number of instances where that's not always the case, or where phages work more broadly than we might have expected. Staphylococcus phages are known to have much broader host range than a lot of other phages. And so some staph phages can kill many phages from all over. That's one example. I'm actually aware of a case where the US Army could not find, some of you may have been even involved in this, the U.S. Army, Walter Reed Army Institute, could not find some phages to kill a particular panel of bacteria. If anybody's on the line, maybe you can remind me. It might have been Pseudomonas. So, and those phages, or those bacteria, might have come from Iraq, but I'm not sure. Anyway, the U.S. Army asked the Kenyans if they could find phages against those, and they found something like 17 phages. And again, those bacteria did not necessarily come from Kenya. Those bacteria came from elsewhere in the world, but phages in Kenya were able to work against them. I do know, for example, there are some Acinetobacter strains in Malaysia that are antibiotic resistant. And I know that the Hungarians in the country of Hungary have found some phages that can work against those. So I think it's going to be phage and bacteria specific, whether we have broad activity or cross-country activity or not.
All right. Yes. Thank you for that clarification again. There's one question again from Joe O'Brien. He's asking, he's saying, great talk. Do current regulations include the use of bacteriophages in combination with the antibiotics?
The regulations are not necessarily what define whether you're going to use antibiotics. It's almost more the ethics. And that is because typically when you test a new drug, you often, most of the time, want to test it against the standard of care. And the standard of care drug in most infections is antibiotics. So most phage clinical trials are done with phages added on top of the antibiotics, where you want to and need to see an improvement above and better than what the antibiotics are doing. And again, a lot of that is for ethical reasons, but then we also know that there are some examples, if you're familiar with the literature, where phages can drive evolution of the bacteria to become sensitive to antibiotics again. And so there might be value in adding phages to antibiotics from a mechanistic perspective. I believe there are a small number of cases where preclinical studies have shown that there is some antagonism between antibiotics and phages. So wherever possible, preclinical studies should be done to see what's the interaction of the antibiotics and the phages before doing that in people.
Yes, amazing. Okay. Maybe to bring back to what we had discussed earlier on Africa approving the compassionate use. Maybe you can highlight to us, like, if Africa was to try and utilize this compassionate use, how would that look like for us? How would the system, let's say, how will the pilot be like?
So I think it depends on each of your countries. I am told, for example, that Kenya has a compassionate use pathway, and that if you apply through their online system, you should have an answer within 30 days. That answer might include more questions that you need to answer before you get approval. I think Uganda might also have a compassionate use pathway. Malaysia does. It comes down to getting approval, I believe, from the Director General within the Ministry of Health. So each country, you need to figure out what is the existing pathway for compassionate use and what is compassionate use called in your country. It might be emergency access, as in the US. I think it might be or expanded access. I forgot what it's called in Australia. It has a different term, something access, special access scheme, I think. So you need to figure out within your country, what is that? And then you need to put together the 6 data points that I talked about. And that means you need to really fully characterize your phage at the genomic level, at the production level, at the bacteria that you use to produce the phage, purification, have outside labs certify the sterility and the endotoxin levels, for example. So if you go through those data sets and put together an appropriate package, you should be able to have that discussion with your regulator. I think the purification step is going to be one of the biggest challenges for a lot of people. I don't think that expertise exists yet in most of Africa, which is why I think it might be valuable to use phages that are initially produced overseas, and maybe even go overseas to learn from those labs how they produce those phages for approval within their countries.
Yes, thank you for sharing that. I think that will be that will be very beneficial for Africa itself, and even for the scientists who are interested in continuing in phage work. They'll be able to even learn from that and be able to come up with better designs to even help us back here in Africa. There's also another question from Joe O'Brien. He's asking that he's saying, if you don't mind me asking another question, I have attended a previous talk where people have discussed the lack of sequencing facilities in labs. Will this be a significant issue with the application of bacteriophages across different places in the world?
I think so, because I think it is, in order to make sure that we're giving safe phages for therapy, we really need to know their genomes. And so I think finding ways to improve that sequencing capability, either in low and middle income countries or through collaborations with other countries. And I'm happy to help sort of make some introductions to possibly facilitate relationships, collaborative relationships to do some of that sequencing, because that is going to be really important. I'll give you an example of why. Cholera phages, Vibrio cholera phages, it has been shown by a group in Bangladesh and out of Harvard in the US that the phages, the bacterial strain that currently causes cholera around the world has a nasty toxin in it that it received, that was transferred to that bacteria by a phage in nature. And so we want to make sure we're not doing something like that in an unintentional way when we're trying to treat patients.
Yes, thank you for highlighting that. There is a very interesting question from Nelson. So the question is that will an ideal situation be sending our phages and pathogen of choice to a lab with the purification expertise to help get this off the ground? So,
it's one way to go. Yeah, I do think collaborations, what I'm starting to think about is how could we get funding specifically to send people back and forth between, say, Europe and Africa to co-purify and to have capacity building, to gain the expertise on how you do that purification, how you do the sequencing if needed. But I think ultimately we do want to see this happening in Africa, but maybe getting it off the ground, not even sending it to them, send the people. I want to see the people going back and forth so that you really get the training, both of the people who are producing the phages and purifying them and of the clinicians. I think it would be valuable to have clinicians going back and forth so that one, you see how it's done, say in Europe, how you treat a patient, but also so those clinicians from Europe or the US or elsewhere could come, say, to Africa and see what's your condition there and how can we help you brainstorm about how to most efficiently do this in Africa. I'd love to see a lot more going back and forth, basically collaboration, because both perspectives are going to be key for making it effective.
Yes. Yes, that is very insightful. And talking about collaboration, which areas do you think Africans should be more focused on currently in terms of phage application?
It's a tough call, and I don't think there's one right answer to that. And I see the question above that, is there a market for phage therapy in Africa? I think it's fair to say, so there is not a great market for any antibacterial drug anywhere in the world, much less in Africa. I can tell you examples of companies where my colleagues have worked in the US, where they made a new antibiotic, it got approved, it worked where other antibiotics were no longer working. They were selling this, it was approved by the FDA, and they went bankrupt because they could not sell the antibiotic for enough to make enough money to keep their company going. And there's two main reasons that are cited for that. One is you can't sell antibacterial drugs, and phages are a type of antibacterial drug. You can't sell them for as much as you can other drugs, like cancer drugs are tens of thousands of dollars, whereas antibiotics are much cheaper than that, and phages would need to be closer to the price of antibiotics. And then second, in the case of some of those companies that went bankrupt, their drugs were held as last resort drugs because they were so good to only treat the patients for whom other antibiotics didn't work. And so they couldn't sell their drug and make money. And that is an appropriate approach from a community perspective, but not from a company making money perspective. And so that's why a number of governments have either individually or together started putting together funds to supplement the antibacterial drug development. And a number of those funds are now being used for phage companies. But that will always be the issue in high-income countries as well as in low-income countries. And this is why I think public-private partnerships, getting governments involved, is going to be absolutely essential to overcoming the AMR crisis in general, whether it's through developing new antibiotics or alternatives like phages.
Right. Thank you for that insights, Dr. Toby. I think I saw a hand from Ellie. Ellie.
Yeah, thank you, Elkanah. So I just wanted to sort of like add some context on the question of getting this off the ground. I'm just trying to understand how willing would these companies that are already well established to purify the phages? Because I understand we have some people who have sort of like a technology on how they can make the phages last longer within the body. And some of these are patented, and most of these companies won't be as willing to just share this information. So, and I really loved the way you put it, how we can reframe the conversation around phages, because it's not about the profit, but it's about stemming the losses that we are losing to antibiotic resistance. So how would we pitch this conversation? Say, for example, us as a training program, we have these students, they're willing to learn, they want to get this product to the market. But then these companies are here, they're saying, we don't want to share our secrets with you. How will you balance that conversation?
Yep. And what I was thinking is actually there's many academic groups that are producing phages that are used for patients. Those are probably more realistically the ones who would be a better place to go learn how to produce, but they're producing phages that are used in patients and that are approved by regulators to be used in patients. So you don't necessarily have to go to a company. In fact, I would recommend going to some of these academic or government groups that are doing this.
Great, thank you.
Like the Belgian military system, I think it would be open. Mm-hmm.
Yes, thank you. I believe this has been a very educative and knowledgeable session that we have had. And thank you, Dr. Toby, again for sharing your knowledge with us, and very insightful thoughts that you have given us. I think when we live here this evening for us is that we are going home very well educated on the regulatory collaborate with the other companies that have already done this to be able to learn as well. And importantly is that we are able now to continue this conversation on phage research and phage work as well. Here with the regulatory bodies, our fellow scientists, academia, and everyone who participates in these matters.
Elkanah, sorry to cut you short, but just before you end the meeting, I saw someone from Africa Phage Forum. Maybe they can say hi. I see they have a comment that I think some of our audience might be interested.
Yes, please.
Yeah.
I have not seen that. Thank you for highlighting that.
So I don't know if it's Nutin or there's somebody with Africa Phage Forum. You can just say hi. We're glad to have you around. Is it me or we can't hear Nutin?
No, not yet. I think I don't know.
Nutin, your audio is not clear. I think you can't hear us.
Yes, but it's good we have Phage Forum here with us, and again, thank you, Dr. Toby, again for being with us here, and us having a very illuminating conversation as well. And importantly, it's I am grateful for your relentless work in advancing phage science where we need it the most. And today's discussion has been a reminder of that phage therapy isn't just about killing bacteria, and it's about reshaping how we think about medicine, regulation, access, and we can copy from countries like Belgium and the UK that that are already testing their flexible models in terms of phage therapy as well. And this brings us to even having the same creativity within our researchers and our scientists here back at home. And we can be able now from that to design a framework that is smarter and even more equitable for us. And importantly for us is that, for us the participants, is that the clear takeaway is that regulation shouldn't be a wall, rather it should be a bridge where we can be able to bring in the discovery and the delivery as well. So bringing in innovation and access together, that is what we need to do. And as scientists, advocates, and even future leaders who are here, is that it is on us to help the regulators understand the science and even helps the science understand regulation. I think I see a hand. So we'll just give 1 minute and then, yes. Femisco, please can go ahead. Mitch. Mitch.
I think you're muted.
All right. Yes. But to what I was saying, it has been a very illuminating session, and thank you again, Dr. Tobi. And we look forward to always learning from you and collaborating with you, and hopefully also building a future where phage therapy is not just a promise but a practice. And thank you as well to our participants for joining in. It has been a very good session, and we are at the beginning of the Phage Week series. We still have 2 more sessions, tomorrow and on Friday as well. So I'll be happy to see you again. I'll be happy to have you around, and come ready to learn, come ready to engage, and yes, continue learning. Thank you. And thank you again, Dr. Tobi.
Nice to be with you all. Thanks for having me.
Thank you.
Just before we leave, like us to. Oh,
I think Nutin is back, Nelson.
okay.
Nutin, hi. Oh, he dropped off again.
I think he might have be gone. Yeah.
Yeah, I think we're out of time, guys. Maybe we can just wrap up and.
Okay.
Yeah, maybe we can have a quick group photo. Then we close the session.
Yes, you can have your cameras on. Right. I think okay. Okay, good.
Great. Thank you again. Hope you all
Yes.
have a good evening.
Thank you.
Bye, everyone.
Bye. So I think the organizing team we can